that I work with Seigo. We work on the monoiron hydrogenase. Today I will talk a bit about the current advances in the maturation of the cofactor. So a bit of an introduction in methanogenesis, in the various steps from reduction of CO2 to methane. One of the critical steps is the reduction of methylene H4-NPT, and that's catalyzed by two enzymes, MTD and HMD. I hope this works. And under Nichols' limiting condition, HMD is the primary enzyme that catalyze this using hydrogen. This is the reaction. Methanol is heteroselectively reduced with an anhydride transfer from HMD in this position. And HMD is a dimeric enzyme that possesses in its active site a very unique organometallic cofactor called the iron GP cofactor. This cofactor is comprised by a GMP moiety, a pyridinol ring, and an iron center. The iron center is further ligated with two carbon monoxide ligands, the cysteine sulfur of the protein, and it's bound to the pyridinol ring by the nitrogen by an acyl carbon ligation that is so far unique in nature to our knowledge. There's also an open site that is proposed to be the hydride binding site. So this cofactor is biosynthesized by the action of 7 different proteins. And here we have the pathway that I will show you step by step. So starting from this pyridinol precursor, we call precursor 1, ACGC is a SAM-dependent methyl transferase that methylates the 3 position, forming 2. Then ACGB inserts the GMP moiety in a GTP-dependent fashion, forming the precursor 3, which is also important because the iron GP cofactor can be light decomposed, and it decomposes into this cofactor. Then this is activated by ACGE in an ATP dependent fashion here in the carboxyl ligand, and then it's bound to ACGF, which is a proposed scaffold for the organic part of the cofactor. From here on, we're not entirely sure how the iron center is formed or where the carbon monoxide ligands come from. We know by a labeling experiment that it can come from either CO or CO2, but we're still not sure why. This is how the pathway looks like, but if you pay close attention, you will see that there are 2 enzymes from the 7 missing, which are ACGA and ACGG. These 2 I will talk about today, and I will show you what we have learned from them. So ACGA is a radical SAM enzyme that has a unique iron sulfur cluster binding motif, CX5, CX2C, so a bit 2 amino acids longer in the first stretch. And it belongs to the biotin and thiamine associated domain containing radical SAM enzymes. If you are familiar with other hydrogenase maturation, you will see that in this group, there are also H2E and H2G. Both of them are responsible for the formation of the iron center in iron-iron hydrogenases. However, even though they present homology to all of this group, we cannot predict the function just by their similarity. So in our lab, we developed an in vitro biosynthesis assay for the iron GP cofactor. In this, we use a methanogen that has the ACGB cell gene deleted. Therefore, this cell cannot produce precursor 3. And all of the other later steps are then halted, and there's no iron GP cofactor. This strain doesn't have any HMD activity. However, we can feed back the purified precursor 3 and recover the HMD activity in a cell extract, in the tube-containing cell extract and some other compounds. And that's exactly what we did. We developed this assay by using cell extract of different Methanococcus maripaludis mutants, in this case, ACGB mutant, and adding several small components, ATP, DTT, sodium dithionide, and S-adenosyl methionine for the radical SAM enzymes or the methyl transferase. And we incubate in an atmosphere of hydrogen, argon, and carbon monoxide. If we remove carbon monoxide from this specific system, the activity obtained is less than 30%. So what would happen then in a ACGA lacking strain? So we had 2 working hypotheses. One, that is derived from the similarities to H2 and H2G, is that this enzyme may be involved in the last step, maybe the iron center or the CO formation. So a deletion of ACGA would impair or create a roadblock in this position and will not produce any RNGP cofactor. And this, if this hypothesis were true, we could not recover any RNGP cofactor by feeding any previous intermediary. An alternative hypothesis was that ACGA is involved in a step before the formation of the first intermediary, either directly or several steps behind. So a deletion of ACGA will block the production of one. However, although there is no RNGP cofactor, we could recover the activities by feeding the chemically synthesized or purified previous intermediaries. That's exactly what we did. So perform the biosynthesis assays with all the small compounds and the different precursors, and we could obtain activity from all of them, so precursors 1, 2, and 3. And there was no activity when we did not add any precursor. For us, that tells us that ACGA is involved in a step previous to the first precursor. In addition, we could incubate the cell extract with no precursor but the purified ACGA, and we also obtain HMD activity. So that tells us that for the first time that the enzyme we're working on, we're purifying, is active, and we can do further assays. But we need to rule out that there's other enzymes that could produce one, maybe ACGA step -1 and -2. So we did is take the cell-free extract and we run it through an Omicron filter with a 3 kilodalton cutoff. We proved that this cutoff had no other enzymes or no other proteins active. And then we did the reaction with the ACGA and SAM, and we did HPLC mass spectrometry analysis. Here on the top, we can see the HPLC MS of the chemically synthesized precursor 1 with an MSAT of 184. And here is the results from the 3 kilodalton filtrate incubated only with ACGA and SAM. And they fit the retention time and obviously the masses. We did this not only Methanococcus malipharulis, but we also use Methanothermobacter marburgensis and interestingly also E. coli. That means that the substrates for ACGA are not methanogen specific. They can also be found in E. coli. This, for me, it's very nice because that tells us that HCGA is really the first enzyme that opens the whole pathway. But we need to discard that it's really this precursor, or it's maybe a precursor that has a similar mass, for example, a similar retention time by coincidence. So we paired both of the enzymes and we run them together. And we see here, this is the product of ACGA. This is a mass spectrometry result. We can see the mass of precursor 1, and when we further incubate with ACGC, this peak disappears and the peak for precursor 2 is shown over time. So that tells us that indeed ACGA generates the first precursor. However, the substrates are still unknown, and that's still an ongoing process to try to find that. So the updated pathway looks like this. So now we have an enzyme that produces from an unknown precursor or precursors this compound 1. And these precursors are found not only in methanogens, but also in bacteria. So just because it's the last enzyme, we postulated that ACGG may be involved in the last step, doing the iron center, the reduction of this to an acyl ligand, forming the CO precursors. And I will show you some evidence of what we're working on with ACGG. So ACGG, at the beginning, we could not even have an homology structure prediction of the enzyme because it was not homologous to any other enzyme. However, by advances in structure prediction by AlphaFold, we can now have a structure and we can study it a bit. It has 2 domains. The end domain is predicted to be a tin barrel that has a very suspicious 3 cysteines and a possible iron sulfur cluster binding motif. Interestingly, this N terminal has a predicted homology with H2, H2, PILV, so the same class of enzymes that I showed you before, also with ACGA. But the main difference is that this enzyme has a C terminal that has a predicted nucleotide binding domain. interesting, this domain has homology to HMD2, which is a paralog of the monoiron hydrogenase that also binds the iron GP cofactor. In the interface between these two domains, there's a strictly conserved cysteine residue that could be hints that a possible iron GP binding. And I will show you a bit further down the presentation, the advances and the models we've done with this. The problem with ACGG is that you cannot express it in a soluble manner in E. coli. So in collaboration with Dr. Cal Costa from the United States, he developed a strain of Methanococcus maripaludis with a his tag ACGG, and we managed to purify it homologously. In black, you can see the as isolated ACGG purified anaerobically, and you can see it has distinct characteristic and distinct peaks, time shoulder about 300 and a peak at 380, a possible iron sulfur cluster 420 peak, and a small one at 600. And this 420 can be reduced with dithionite, although slightly. We did the biosynthesis in a delta ACGG strain. And interestingly, this we could not recover the activity even when we added precursor 3. That means that indeed the roadblock is in the last step of the biosynthesis. When we don't add any precursor, but we add our enzyme, we obtain a tiny bit of activity. So some of the precursors and the substrates are present in the cell-free extract. Of course, when we add precursor 3 and our enzyme, we can recover a lot of the iron GP cofactor activity. And interestingly, we can skip the carbon monoxide in this case and only incubate under hydrogen, and we have around 80 to 90% of the activity that without carbon monoxide. So this was interesting because for the first time we could remove carbon monoxide as a substrate, and it means that it hints that adding extra ACGG to the system produces the CO ligand. But analyzing the iron GP cofactor by mass spec is a bit hard. So what we did is all of the radical SAM enzymes have a common mechanisms that they produce 5'-deoxyadenosine as a product. So, and this is stimulated when the substrate is present. So when we did the 3 kilodalton filtrate and incubated with ACGG, we see a big increase over time. So this is 1 hour, 3 hours, and 6 hours, and over time of production of 5'-deoxyadenosine. Common radical SAM inhibitor, SAH, inhibits this reaction. But interestingly, when we incubate these conditions, but in an atmosphere that has 50% carbon monoxide, this reaction is inhibited. This is news to us, and to our knowledge, it has not been shown in any other radical SAM reactions, which is very interesting, a radical SAM reaction inhibited by carbon monoxide. We explored this further. So because of the homology to height G, we thought that maybe it's an amino acid that could be the one like tyrosine to produce carbon monoxide. So we incubated ACGG in a solution of Cas amino acids, and we did a myoglobin assay and measured the carbon monoxide production via the change in the spectra. And indeed, we could obtain some carbon monoxide, but when we removed the carbon monoxide on only buffer, we also obtained some activity over time, which made us a bit suspicious because, well, the Cas amino acid solution was also done in this buffer. The buffer was 50 millimolar tris. It was a bit suspicious, so we did a concentration dependence production of carbon monoxide with only tris in MOPS buffer, and we see that indeed there is a concentration dependent, ACGG dependent production of carbon monoxide only with tris. And this, well, this is a bit an ongoing research at the moment. We are not sure if it's really tris that is the carbon monoxide buffer. That would be very unlikely. We cannot see it chemically, how could it be? It could also be a contaminant that's present in most tris solution, because if you see here, this is 50 millimolar tris, but only around 0.6 micromolars of carbon monoxide are present. So it's in the range of a possible contamination. And common contaminants of tris are formic acid or other 1-carbon, 2-carbon derived compounds. So it could be something, but we need to explore this further. Because of the homology of ACGG with HMD2, our postdoc Yu-Chong Tseng did a study on the possible binding of the iron GP cofactor to ACGG. And here we see the alignment of, in blue, the ACGG model and the crystal structure of HMD2, so the monoiron hydrogenase paralog. And they fit relatively well. But also the residues that binds the cofactor are in very good agreement with them. And here, so what he did is superimpose the iron GP cofactor and then did minimization of the energies to try to find it, to bind it to ACGG, and indeed it binds very well. And this conserved cysteine directly points to where the iron could be. To further study this, he purified ACGG and then did a reconstitution experiment when he incubated ACGG with a purified iron GP cofactor. And indeed, the protein binds the iron GP cofactor. So in black, we can see the spectra similar to what I showed you before. In orange is the spectra of the iron GP cofactor, and in red is the spectra of both of the FAGP cofactor bound to ACGG. And you can see it looks like the same spectra, just a bit higher. And interestingly, this iron GP cofactor could not be washed away by exchanging buffers. So there's a very strong binding in between the cofactor and the enzyme. So we're exploring a bit further this. So as a short summary, we explored the functions of ACGA that went from an unknown radical sum enzyme to a known H2ase homolog. However, even though they're homologies, it catalyzes a very different reaction, which is the formation of the first pyridinol precursor, although the substrates are still unknown. We have some ideas from the substrate. We discarded that is an amino acid. We did 15 unlabeled amino acids, and we incubated with ACGA, even in cell extract. Maybe it's a second or a third step afterwards, but we did not find any label in the precursor 1 form. So we're still hunting for this substrate. And for ACGG, we know it's a novel radical sum enzyme that catalyzes the formation of carbon monoxide from another unknown substrate. And it has a mechanism that involves inhibition by carbon monoxide that we have not seen before. Again, and also it needs to catalyze the full assembly of the iron GP cofactor. And because it has this binding motive, it also may serve as a scaffold and delivery function to the final APO enzyme. We're still exploring this at the moment. We're trying to characterize ACGG further because the yields we obtain from the homologous expression are very low, and we still want to do a spectroscopy with them, so we're just purifying. I want to thank my whole group, Agashima, especially Seigo, for giving me the opportunity to run my PhD, to do my PhD with him. Also the collaborators, Cal Costa, Yon Lei, that provided the Methanococcus maripaludis mutants we use, and all of you for your attention. Thank you so much.